So much for the invitation to speak to you today. I was hoping to come to Hawaii, but this, of course, is a much easier way to connect with you and to share the research that we do in my group. Yeah, I'll start with a little bit about the research center, where I am, what my program does, and then I will slowly get into how to deal with stowaways on your plants. So the Violent Research and Innovation Center is an independent, not-for-profit organization that was launched in 2007. However, where we are, there has been an experimental research station already for more than 100 years. So we serve the growers, the companies that supply the growers, and our goal really is to make things better for them. Economic viability, sustainability, and competitiveness of the horticulture sector. So horticulture not only includes greenhouses, but also many outdoor crops. We really are focused on delivering results, products, solutions, services, through an integrated and collaborative approach. So Violent has 5 research programs, of which I'm leading the first one, biological crop protection. But we also have a group that looks at consumer sensory and market insights. We have a group that does crop breeding. We call them the Crop Enhancement and Adaptation Program. We have a group that looks at how to best grow plants, so plant responses in the environment. And then we have a group that looks at any technological solutions, including automation, called the Horticultural Technology Solutions Program. So a bit more about biological crop protection here at Violent. We are really all about enabling and accelerating the introduction and use of biological plant protection strategies to reduce the negative impacts of pests, diseases, and a little bit of weeds in horticultural crops. So I'm an entomologist by training, but our group also includes a plant pathologist so that we can work together on all the pests and diseases in the crops. We actually work on new products, so new biologicals. You see a good example at the bottom left. That red mite is actually a new biocontrol agent that was commercialized a few years ago now. I don't know if you have it on Hawaii because you're not part of the mainland, kind of like North America. But who knows, maybe it will make its way onto your part of the world as well. We look at integrated pest management solutions. The middle plants, the chrysanthemum you see is attacked by thrips. Thrips have kept me employed for more than 20 years. And I'll talk a lot about thrips today. And we'll also look at the environmental performance and safe working conditions in our research to deliver on that, all through biological tools. So a bit of background from my corner of the world. Ontario, Canada is, we're located pretty close to Niagara Falls. So that kind of hopefully situates you a little bit, the Great Lakes area. And we have a very big, for us, greenhouse industry, including greenhouse floriculture. And every 3 to 4 years, we ask them, what is your use of biological control? And this greenhouse schedule that you see from between 2014 and 2022, actually you see that almost all growers are using biological control. And the main reason for that is pesticide resistance. So we have pests that we just don't have effective products for. The other reasons that growers use biological control is no re-entry intervals or pre-harvest intervals, worker health and safety, and just because it is effective for them. The very small percentage of growers that don't use biological control is mainly because biological control is not compatible with the pesticides that they use. They don't know how, or they just don't have a lot of pests. So over the past 5 years, 70% of growers actually have reduced their pesticide use and increased their biological control use. So that's a really good metric for us to report on. The most problematic pests were Western flower thrips, 2 spotted spider mites, aphids, and mealybugs, and each accounting for about 5 to 10% of crop losses. So that's kind of the picture of pests in ornamentals. So when I said that thrips have kept me employed for the past 20 years, it's because thrips were first a roadblock to the adoption of biocontrol strategies. The growers had to spray against thrips, so it was very hard to actually make biocontrol programs work because a lot of these pesticides were not compatible. However, thrips were one of the first pests in floriculture where basically we had no products that worked. So the growers had to do biological control. There was a big research effort on biological control of thrips that I participated in. And as of now, probably more than 70% of a grower's biocontrol budget is spent on thrips in greenhouse ornamentals. So when you spend that much money on biocontrol of one pest, you have to make, nothing can interfere with that. So they had to make biocontrol of other pests work as well, because a lot of these chemicals are not compatible with biological control of thrips. So it became basically the cornerstone of biological control in Canada. So at some point we were like, okay, I think we've got it pretty much figured out. We know how to do biocontrol of thrips and done a lot of research. So we should know what we're doing, right? And then our last few surveys actually came back saying that thrips are still listed as the most challenging pest, most crop losses, and growers were not really happy with the biocontrol program against thrips. Also, about half of them, or more than half, started experiencing more frequent or severe thrips outbreaks in the past 5 years. So something changed. And that's when we started looking at some really smart person, not me, came up with the question like, are we still looking at Western flower thrips? Because of these increased thrips outbreaks, but no one knew because no surveys, thrips surveys had been done since like the 80s. And thrips are so tiny, as you can see on this tip of the pencil. Like, it's easy to just assume that the species you're dealing with is Western flower thrips. So in 2016, my colleague Sarah Jendrisic from the Ontario Ministry of Agriculture did a big survey and found that, yes, there's still a lot of Western flower thrips, 65%, but about 1/3 were actually onion thrips. And the Latin name for onion thrips is Thrips bassi. And then a few other thrip species, like we always have some echinothrips and other species. But the onion thrips were kind of unexpected. Then we started looking into different crops as well, other greenhouse crops, so including vegetables like tomatoes, cucumbers, peppers, eggplant. Now have quite a few strawberry greenhouses. And of course, here in Canada, the cannabis industry is legal as well. And we found a few surprises. Onion thrips were actually present in a lot of tomato greenhouses, and it was the dominant species in cannabis greenhouses. Now, this survey did not include all the greenhouses, so this is just a snapshot, but it illustrates that onion thrips actually can make up quite a big portion of a greenhouse thrips population. And we see that the onion thrips, biocontrol of onion thrips has to be slightly adapted to be actually effective. We also find an increasing number of non-native thrip species on imports. So the onion thrips and the western flower thrips, we consider them naturalized. They've been in Canada for a long time. Onion thrips actually come from outdoor crops into the greenhouses, and western flower thrips come in either on plant material or from outside. But these new exotic thrips are increasing and can include this whole list that you see here on the right side in the green box. And the reasons for more thrip species coming in is increased global trade. Also during COVID, everyone wanted to have house plants. So the tropical foliage plants came in with some interesting thrip species. We have less and less harsh pesticides to actually use on our crops. Climate change also expands ranges of thrips. And when you start looking, you will find a lot of things as well. So we developed a key. Actually, this was developed by someone from my lab, Ashley Summerfield, and again, this a researcher from the OMAFRA ministry, to make thrips identification actually work, because they're so small. And traditional scientific keys are very technical and require quite high-powered microscope. So they developed a key for growers and consultants that only requires like one of these dissecting microscopes and only includes the species in Ontario. So this QR code will take you to the key, but in Hawaii, you probably will have to develop your own key because you will probably have different species than we have. So I would not advise using our key where you are, but it's a good idea to maybe develop one of your own. So here's kind of the program that a lot of growers have been using for western flower thrips in chrysanthemums. And it includes from left to right, it goes from little cuttings in chrysanthemums to the flowering product. And it starts with biocontrol, with Diiping Cuttings, which I will spend the rest of my presentation about. A lot of biocontrol also once the cuttings come out of the misting house, including nematodes, predators, some biopesticides, and then mass trapping as well. And then at the end of the production cycle, when if there's still thrips left, then growers start considering using a chemical pesticide, because by that time, the thrips may actually be susceptible to that particular pesticide, and it will not interfere with the biocontrol program anymore, because the plants are going to leave the greenhouse real soon. So really one of the most important parts of integrated pest management programs in flowers, but in other crops as well, is just to start with no pests and to keep the crop pest-free as long as you can, so stay clean. And we found that in floriculture, these unrooted cuttings that we receive at the beginning of to start our plants with can be an important entry route for pests. And to make it more doubly problematic is that these incoming pests may already have been exposed to a lot of pesticides at the propagator, because the propagators don't want thrips on their plants as well. So these incoming pests may already be resistant to multiple pesticides. So the key is to establish a really strong biocontrol program at the beginning, and to try and start as clean as possible. So for that, we developed these cutting dips. So how many pests and which pests actually come in on cuttings? We have different crops here that we looked at. Poinsettia cuttings often come in with white flies. Infestation levels may vary year by year, but it's mostly these really tiny eggs and nymphs that are hard to see. It's many different cultivars, so it's not cultivar specific to up to an extent. Some cultivars have more, but then others. And it's often this pesticide-resistant species or biotype of whitefly. In this case, it's Bemisia tabasi, or the sweet potato whitefly. Chrysanthemum cuttings often come in with western flower thrips or spider mites. And it can be quite high numbers, and a lot of the different cuttings are infected. Not a lot, like 1 to 2 thrips per 20 cuttings, or up to 118 mites per 20 cuttings. That's a lot. Thrips, it's mostly eggs and larvae. Spider mites is all developmental stages. And again, a lot of different cultivars have these pests hitchhiking. So what happens if you start already with a few pests on at the beginning of your crop? Well, inevitably, their populations will grow rapidly. And biocontrol agents are not likely to catch up right away, because the pests have a head start on the bios. And the bios may also be affected by pesticide residues on the cuttings which will make them perform less well. Now, the use of pesticides may not work because the pests that come in may already be resistant. And it will also interfere with any bio program that you will probably want to do later on in the year. So the options are really, yes, you can scout incoming propagative material, but these really tiny pests are hard to see. So it's probably safe to assume that the incoming material is infested. This can be based on experience or records from previous years, that can already indicate that there are maybe pests present. But luckily, there are things that you can do. So dipping is one of the things which will kill a big part of the pests that come in. Also, start as soon as possible with the biocontrol program. And then if you have to use any sprays, then try to prioritize the soft pesticides that are compatible with the bios or the biopesticides. So what are cutting dips actually? Basically, you take the cuttings out of the bag and spread them loosely in a perforated tray, and then immerse them completely into the dip solution. Really, the goal is to get complete coverage of the cuttings. Excuse me. The products we investigated as dips were biopesticides or reduced risk products such as oils or soaps, because those are generally just much more compatible with biocontrol that you want to do later. Have less issues also with pesticide resistance, and leave minimal residues. And then after dipping, the cuttings are stuck in the substrate and just grown as usual. A tip is, if you're dipping cuttings, part of the pest will actually wash off. So if you collect the dipping solution and kind of filter it through a filter paper or a coffee filter, you can actually see some of the pests that may have washed off the cuttings, to give you kind of an idea of if anything was actually present. So in the coming slides, I will talk about some of the research that we did at Vineland, efficacy versus phytotoxicity of certain products, the compatibility of dips with biocontrol, show you some commercial greenhouse trials, and also talk about one of the main concerns that plant pathologists often have with dips is the risk of disease transfer through the dip. And then the question also comes up, does dipping actually carry any cost benefits? So let's have a look at what we found. We tested a lot of different dip products. For whiteflies, Bemisia tabaci on poinsettia. For thrips, western flower thrips on chrysanthemums and various bedding plants. And for spider mites on beans. And on the right, you see the different products. In the States, they may have slightly different names, but the red ones are basically the oils, either mineral oil or vegetable oil. In blue, you see the soap. And then in green, you see the biopesticides, such as the entomopathogenic fungi or nematodes. And this table briefly summarizes both the efficacy and the phytotoxicity of all these products and combinations. So you see in the most left column, you see the products and the combinations that we used. For example, we mixed soap and entomopathogenic fungus, Buitenhuis, together, and actually got really good efficacy on whiteflies. So then in the next few columns, you see the different pests that we dipped against, Bemisia, thrips, and two-spotted spider mites. And then you see them again because we also tested it on sensitive crops for phytotoxicity. So the green, the percentages in green, you can actually see that these products were actually really effective against these pests. While on the right columns, you see some yellow warning triangles that actually mean that these dips were phytotoxic for sensitive crops like poinsettia, mini rose, petunia, and osteospermum. So you can see there is at least one or two products or product combinations that will work for each of the tested pests. So the good news is there are lots of products that will work. With soap and oil, you do have to be careful because first of all, the spray rates are much higher than the dip rates. We had to go lower rates for dipping due to phytotoxicity. So it's a good idea to always test for phytotoxicity first. Also, mineral oil kills thrips eggs. And these thrips eggs are actually inserted into the leaf tissue, so they're very hard to kill. But the mineral oils such as sulfur X and landscape oil actually killed these eggs. Also, a few products like veg oil or entomopathogenic nematodes that were not very effective as dips, with the caveat that entomopathogenic nematodes may be very effective when you're dipping rooted plugs so that they get into the substrate. But on the foliage only, the entomopathogenic nematodes did not perform very well. So now, of course, you dip your cuttings, and then you have to continue with the biological control program. So we tested the efficacy of this kind of sequence of treatments, again, on whiteflies and on thrips. Whiteflies on poinsettia and thrips on chrysanthemums. And I'll spare you the experimental details and go straight to the results. So for whiteflies, in this graph, you can see over the course of several weeks, week 1 to 7, you can see that the number of whiteflies per plant increased when we did no dip, but only biocontrol. So that's the blue dotted line. You see that we go from basically very few whiteflies to over 15, 16 whiteflies per plant with biocontrol only. When we started dipping in soap and Buitenhuis, in this case, you can see that the number of whiteflies in the yellow solid line stayed pretty low, but eventually the effect wore off and the whiteflies started to increase again. The yellow dotted line is actually the combined treatments of dips and biocontrol. And you see that there, the whitefly stayed consistently very low. So dips and biocontrol resulted in the best whitefly control. We tested this with whitefly parasitoids like Encarsia formosa, but also did it for with a predatory mite, Amblyseius swerskiae, and got similar results. When we did the same experiment with thrips on chrysanthemums, again, we see the different treatments. So in this case, we also had a treatment where we did nothing, which is the solid blue line. And you can see the thrips just explode on the plants over the course of 4 weeks. The dotted blue line is when we did biocontrol only, and we released predatory mites. And you can see that the predatory mites did have an effect on thrips, but the thrips still increased over the course of the time. Now, when we did dips in either an entomopathogenic fungus, Bovaria bassiana, and the trade name is Buitenhuis, or in landscape oil, so these are the red and the yellow lines, you can see that when we combined dips and biocontrol together, again, we got a very good thrips control. And the red line is actually behind the yellow line, and that's why you can see it very well. We had a surprise infestation of the cuttings with spider mites. So this turned also into a spider mite experiment. And here we see very similar results. The oil dips reduce spider mites really well, while the Buitenhuis, so the entomopathogenic dips, did not. Also, the biocontrol agent that we released, swerskiae, can prey on spider mites, but it definitely prefers thrips when present. But we could definitely say that at least the oil dips worked quite well. Also, again, in combination with the biocontrol agent, which is the yellow dotted line. So you can see it's really all the spider mites stay low. Of course, you can prove it in the lab, you can prove it in the research greenhouse, but then we had to, of course, go to commercial greenhouse trials, which we did. And again, we saw that the undipped cuttings generally had more whiteflies, and we did not see any effect on plant growth. So again, that was good news and really convinced a lot of growers that this was a good idea. I already mentioned that one of the concerns of plant pathologists is that the dips could potentially transfer pathogens from infected cuttings to healthy cuttings through the dip. So before we could recommend this strategy to growers, we really wanted to see if this was the case. So we did an experiment. First, we started to look like, do we see soft rot causing bacteria called Arwinia or Pectobacterium in dip solutions at commercial greenhouses. So what we did in this table, you can see 4 different greenhouses, A, B, C, and D. And the source water was all clean, very nice. Before even dipping one cutting, we saw that there were, in some greenhouses, there was already some bacteria there, which was probably because the tank hadn't been cleaned very well. So that's definitely a pointer there that you have to clean out the dipping tank before and after you use to prevent any buildup of pathogens. The other thing that we saw is definitely after dipping cuttings, we could find quite high numbers of these soft rot causing bacteria in the dipping tank, in the dipping solution. But we never really noted any disease in the dipped cuttings. So the question was like, will very high levels of Pectobacterium actually cause healthy cuttings to become infected? Which we tested in the next experiment. We had several treatments where we had very low bacteria concentrations to enormously, unrealistically high bacteria concentrations. We did 2 different varieties of Poinsettia, And we replicated this several times to see if we could get consistent results. So what we saw was that there was definitely a variety difference in susceptibility. So the variety prestige red developed more disease than the freedom white. And not surprisingly, if we had really high numbers of bacteria in the dip solution, we did get some cuttings that were previously healthy that got disease. However, it never got really above 20%. And this was really in the highest number, the unrealistically high cutting dip solutions. So the risk of Pectobacterium transfer in unrooted cuttings through the dip is pretty low. And lastly, we did an economic analysis where we compared biocontrol alone, biocontrol and dip, and pesticide treatments. And we basically saw that there were very little differences in the expected net benefits. It was just a few cents higher than with biocontrol alone. Chemical control was slightly cheaper, but again, I have to emphasize the risk that the pests are already resistant against the pesticides. So this economic analysis did not include that risk. So overall, it just makes sense to do dips as an insurance to reduce the risk of pest outbreaks. So to sum it all up, I know I presented a lot of information. These cutting dips of unrooted cuttings can effectively suppress whiteflies, thrips, and spider mites. These dips that we tested are compatible with biocontrol agents and can also improve the overall biocontrol program because we start with lower pest populations. The risk of disease transfer of this bacteria was really low in poinsettia, so it's an acceptable risk as long as you do very good sanitation to reduce the numbers of the initial load of bacteria. And dips basically make sense economically. So as a result of all this research, dipping has been added to the product labels of many products as an application method. In Canada, we have 4 products that can now be used as dips. And in the US, I'm very jealous, you even have more products, including Buitenhuis, wettable powder, Tetracurb, Impede, insecticidal soap, Hexagon, that's a pesticide, EpiShield, and Lalgard M52, which again is an entomopathogenic fungus. The uptake by growers has been phenomenal. About 3/4 of growers in Ontario dip their cuttings, which is an amazing result. And there are many options for further research because we could not test all products and all pests. So and there is a lot of potential for further research. Now, coming back to some of these invasive species, of course, a lot of these come in on plant material. So for one of them, Thrips parvispinus, which is an increasing pest now here in Ontario as well, we repeated our dipping experiments and found that a lot of these products, including Buitenhuis, landscape oil, and Sulfol X, actually worked for parvispinus as well. So that was a really promising result. Now, if you want to do cutting dips and you're wondering how to do it, we did produce a little video because a video is so much better than just hearing me talk about it. You can see actually what the important pointers are. So if you go to a YouTube channel called Greenhouse IPM, without a space between Greenhouse and IPM, you will see this video and you can review the methods at your leisure. We use perforated trays, but some people actually use these deep frying baskets or even a colander for dipping. So that's our all options. If you want to dip and there's no information yet on phytotoxicity or efficacy, you can do some testing yourself. I think it's important to start on small sections first, not more than like 10 to 20 cuttings, and do it at least a few times so that you can see if it's the same effect every time. Also compare it to water or non-dipped cuttings so that you know if you don't dip, and if you dip, if there's any difference in the health of your cuttings. And take good notes. So make sure that you know what you're measuring and that you write it down so that you can see it later. Very briefly, there are some other technologies that are very interesting. Like I came across this company that developed actually a steam chamber. They call it a plant sauna to disinfect cuttings in a similar way, but instead of immersing the cuttings, they use steam treatments of a certain duration and a certain temperature. There's also this local company close to where I am that uses a combination of UV light, ozone, and hydrogen peroxide treatments to disinfect. So we're going to work with them, hopefully, to see if this is an alternative to dipping for certain plants. Now, to wrap it all up, of course, biocontrol is only one part of an IPM strategy. To get effective pest control, you also have to look at how do your production practices affect pests and biocontrol agents, such as fertilizer or screening, and also choose resistant varieties or improve a plant's ability to defend itself. And with that, I think I'm going to skip the last slide, which is just a bit more of these principles. And I'll open up the floor for questions. And I thank you very much for listening. I hope you learned something and that I can help you develop maybe some of these products and strategies for yourself. Thank you very much.